Hello everyone, I welcome you to the biology class once again, and I must appreciate you for joining me again in this class, and I hope you are really learning. I will implore you to please like the video, share the video with your colleagues, and also subscribe to my channel so that anytime I upload educational content, you'll be able to get a notification and be able to learn. Okay, so let's get started. In this video, I'm talking about development of new organism. Okay, development of new organisms. I'm going to be talking about animal and then talking about plants. Just like in my previous video, I'm going to be using certain picture to explain development of new organism. Let's get started. The first one I'm talking about is stages in the development of toad. Okay, when you talk about the development of toad, as you know, toad is an amphibian, and the development of toad has three major stages. As apart from the courtship stage, you have the egg stage, and then you have the young tadpole stage, and eventually you have the adult stage. Okay, so the first stage actually is the courtship stage. As you can see here, you have both the male and the female toad, and like I said, the first thing that will happen is a courtship, and in the courtship the male will make a loud quacking noise, okay, in order to attract the female, which is already pregnant with or we can say it's already swollen with eggs. Okay, let me use the word swollen with eggs. So the male toad will later climb on the female's back and hold the female tightly or firmly with the thick parts on his thumb. So as the female will lay eggs, the male then will go ahead to pour the sperm or release the sperm on it, and that is the egg that is formed here. So we can say this is the first, which is the courtship stage, and then the egg stage, the second stage. Okay, so the egg at the egg stage is basically laid around a string of jelly. Okay, they are surrounded by a string of jelly, and at that particular point, mitosis will take place so that the fertilized egg can develop into an embryo. Okay, so the embryo will still be feeding on the egg yolk at this particular stage. Eventually, you have the egg hatched, and then the tadpole is released, okay, from the egg. Now, the series of changes that will take place from this particular tadpole, which I term number 3 here, up to this point is what we refer to as metamorphosis. Okay, metamorphosis actually happen in this organism because the changes that takes place from this young tadpole to the adult, each of those stages you have different features being developed by the organism. And then, okay, so the next thing you have here, you can see, I will term this number 4. Next thing you have here is the external gills develop, so that the tadpole will be able to make use of the external gill for respiration. And then you also have the hind limb emerge, okay, for the organism. And then you come to this point, I will term number 5, where you also have the forelimb. Okay, you have the hind limb here, and then you have the forelimb here, and then over here you see that the tail has disappeared. Okay, and already you have a young tadpole, okay, with the 2 limbs, and then you have a fully grown toad here, and that's what we refer to as a metamorphosis, series of changes that take place from egg to what, to the adult toad. And that is what happened in the development of amphibians, in this case the toad. Okay, so that will lead me to metamorphosis in insects. When you say metamorphosis in insects, metamorphosis actually simply means the series of gradual changes in both forms and shape that bring about a transformation in insect from fertilized egg to adult, from fertilized egg to adult. So those series of changes they are referred to as a metamorphosis. You can see even in this uh, f- uh, the stages in development of toad that I've talked about earlier, you see that over here you have some features, and then as additional feature here, that's the hind limb, additional feature here, that's the forelimb, additional feature here, the tail that disappear, okay, and over here you see that the toad is already fully developed. So that's what we refer to as metamorphosis. So we have two types of metamorphosis in insects. We have the complete metamorphosis and we have incomplete metamorphosis. When we talk about complete metamorphosis, that means series of gradual changes that takes place in insects from fertilized egg to larva, and then to pupa, and finally to the adult. And this occurs in some type of insect. Okay, so in complete metamorphosis, you have the egg form, and then you have the larva, and then you have the pupa, and lastly you have the adult. You have the adult. Okay, you have the adult. Now, to talk about the types of insects that make use of this complete metamorphosis, or that develop uh, in these four different stages, I'll use the acronym B3HMW. Okay, the first B here is beetle, the second B butterfly, and the last B is bees. Okay, H is housefly, M is mosquito, and W is wasps. Okay, so all these six type of insects, they pass through what we can refer to as complete metamorphosis. Okay, and you can see a very good example here. This is housefly, the egg stage. From the egg you have the larva. By the way, in housefly, the larva is also referred to as maggot. Okay, maggot, referred to as maggot. Okay, so and then you have the pupa, and finally you have the um adult stage. So that is for complete metamorphosis. Now, in the incomplete metamorphosis, the insect will go through three stages. Okay, unlike the complete where they go through four stages, here you have three stages. The first stage is the egg, followed by the nymph, and then finally you have the adult. So you have the egg as a first, and then you have the nymph as the second, and finally you have the adult. You have the adult. Sorry, this writing is not, the screen is not really cooperating. Okay, so you have the adult stage, and the type of organism that or the type of insect that undergoes the type of metamorphosis, I will use the word TAP, 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 GC, tap, GC. Okay, so I say the first T here is termite, the A here is aphid, the P here is praying mantis. Okay, the the G here is grasshopper, the C is cockroach, and then L is a locust. Tap GCL, tap GCL. Those organisms undergo incomplete metamorphosis, made up of three major stages. You have the nymph, and then, oh sorry, you have the egg. Now, in this case of cockroach, the eggs are laid in this particular case, and you call this case ootheca. You call this case what? You call it ootheca. Okay, this particular case is called OOTHECA. ootheca. That's singular. Okay, ootheca. And then when the egg is hatched inside the ootheca, the case will break and then will give rise to nymph. The nymph are usually very colorless at the beginning, and then as they begin to develop, they become more brownish, and that's why you have the adult cockroach. So that is for incomplete metamorphosis. Okay, so I hope you are getting it. If you have questions, you can drop in the comment box and I will come to provide answer to those questions. Now let's talk about development of new seed. We are moving on to the plant now. When we talk about development of new seed, remember that the first thing that happens is pollination. And for reproduction in flowering plant, the first thing is pollination. What is pollination? The transfer of pollen grain, okay, from the anther to the what? To the stigma of the flower, either of the same flower, another flower. That's what you refer to as a pollination. Now, after pollination has taken place, the pollen grain will now move down from the stigma through the style. You can see this is the style, move down to the style, okay, and it's heading to where? It's heading to the ovary, where you have the ovules, and inside the ovules you have the egg, just like you have the structure here, okay. So just laying the background or the foundation for you to understand that what I'm about to talk about actually starts from pollination. Okay, so this is the pollen grain. This is how it looks like. The external part is referred to as the exine, while the internal part is referred to as the intine, and then there is nucleus. Okay, you have the nucleus there, and then you also have the cytoplasm. Okay, so this is the way the pollen grain looks like. Now, when the pollen grains land on the stigma, it will absorb a liquid which we refer to as a nectar. When it absorbs the nectar, okay, it will begin to swell. Okay, we begin to swell, and the swelling will make it to eventually break. But before then, you see that initially you have just one nucleus here. Okay, but by the time you by the time the pollen grain absorb nectar or the sugary liquid, it will then begin to develop, and it will give rise to two types of nucleus eventually. Okay, you have the tube nucleus, okay, and then you have the generative cell which produce generative nucleus. Okay, so then when the what's it called now, the pollen grain begin to um absorb the nectar, it will then break and begin to grow tube. Okay, you can see the tube nucleus will begin to grow a tube, and the tube is growing down inside the what? Inside the style, heading towards the what? The oval. Okay, just like you can find it, you can see it here. Okay, so the tube eventually, okay, goes inside, and you can see you already have two type of nucleus. You have the tube nucleus, and then you have the generative nucleus. Okay, so the generative nucleus eventually give rise to the male gamete. Generative nucleus divide into two nuclei, okay, and those male nucleus becomes the male gamete. So as the male gamete is being formed, you can see that here, okay. The pollen tubes will then grow into the oval through the micropyle, and then the ends of the pollen tube will burst. This is the end of the pollen tube. This is the end. It will burst. So when it bursts, one of the male nucleus will fuse with the oval to form zygote. Okay. So when one of the male nucleus has fused to form zygote, that is the first fertilization. And then the second male nucleus will then fuse with the secondary nucleus to form a triploid cell. which will give rise to the endosperm nucleus, okay, and that will produce the endosperm. I will show you the endosperm shortly, and that is what we refer to as the second fertilization. Okay, so flowering plants undergo what you can refer to as double fertilization. Okay, the first fertilization give rise to the zygote, and the second fertilization give rise to the what? To the endosperm. Okay, so this is the seed that is formed eventually. Okay, actually, after fertilization, the ovule, this ovule you are looking at here, will develop into seed, while the ovary, which enclose the ovule, will develop into fruit. Okay, so having known that, this is what you have eventually. The seed that is formed from where? From the ovule. You know what the pericarp is? The pericarp of a fruit made up of the mesocarp, the endocarp, and the epicarp. Okay. Then the endosperm. Don't forget we said the endosperm is formed from the second fertilization. Okay. So this is the endosperm now, and you have the cotyledon here. Okay, the cotyledon basically consists of food. Okay, that's the storage of food, where food is stored, because the seed is a living thing and it will need to grow. So you have a stored food in the cotyledon. Then you have the coleoptile. The coleoptile actually protect the emerging shoot, the shoot that will grow outward or that will grow upward from the seed when planted. So the coleoptile will protect the what? The emerging or the growing shoot. Then you have the plumule, which develop into the shoot. You have the radicle, which develop into the root, which will grow downward. And then you have the coleoriza. The coleoriza or coleoriza will help to protect the emerging root from the radicle. I hope that is clear. Okay, so this is okay. The one we have here is a monocot seed, and this is a maize seed. Here you have a dicot seed, and this is a bean seed. You have the position of the radicle. The radicle, I told you, will develop into the root. You have the micropyle. The micropyle actually is the structure that allow water to enter into the seed. You have the hilum. The hilum control the contents of the water in the seed. You have the tester. The tester is also referred to as the seed coat. Okay, that's like the covering of the seed. Okay, now this is a complete seed. That's a complete, the external appearance of a complete dicot seed. Here you have still a dicot seed, but the tester has been removed. Okay, when the tester is removed, you can see the cotyledon clearly. Okay, and then the radicle, which develop into the roots. Okay, so here you have just one part of the cotyledon. This is the dicot, like I said earlier. So this is just one part of the cotyledon. You can see clearly the plumule that develop into the shoot, okay, and the radicle that will develop downward, forming the root. And then you have one part of the cotyledon. Okay, now let's move on to how seed germinates. When we talk about seed germination, we're referring to the process by which the embryo in the seed will become activated and begins to grow into a new seedling. Okay, and of course, you know, after the zygote is formed, mitosis will take place for the zygote to become an embryo. I talked about that during my when I was talking about the toad, development of the toad. Okay, so mitosis will take place for the zygote to develop into what? Into an embryo. Okay, so that at that point when the embryo is formed, when the seed is planted, or when conditions that are necessary for germination are fulfilled, the seed will then begin to grow. So the first one I'm talking about, we have two types of germination. The first one is hypogeal germination, and the second one is epigeal germination. When you talk about hypogeal germination, Okay, what is hypogermination? It's a type of germination in which the cotyledon or cotyledons remain inside the seed coat and they do not come above the level of the soil. This is a very good example of hypogeal germination. You can see that this is the level of the soil. This is the level of the soil. Okay, you can see the seed. Okay, you can see the seed planted, and you can see after the seed has been planted, see the root, the radicle growing downward as the root, and then see the shoot growing upward. Okay, the shoots growing upward. See the coleoptile that I said will protect the emerging shoot. Meanwhile, the seed remains below the soil. That's hypogeal germination. The seed remains below the soil. Example of such type of germination is found in all monocotyledonous plants like maize, like guinea corn, like millet, like wheat. They undergo hypogeal germination. Okay, and the other type of germination is the epigeal germination. In epigeal germination, the cotyledon or cotyledons will come out of the seed. Okay, will come out of the seed coat above the level of the soil. Again, this is the level of the soil. Okay, that's the level of the soil. The seed is planted. You can see the radicle growing downward, and then you see the seed coat. You see the seed coat. Okay, and then what do you see eventually? The cotyledon is carried above the soil. Look at the cotyledon. Unlike what you have in hypogea, the cotyledon remain below the soil. The cotyledon remain below the soil. So in epigeal, the cotyledon is carried above the soil, and then the first leaf emerging from the midst of the cotyledon. You call that the first foliage leaves coming out of there. Okay, you can see before the cotyledon eventually drop off. So it is usually carried above the soil while the root grows downward. Okay, so that is epigeal germination. And finally, let me quickly talk about let me talk about the difference between epigeal and hypogeal germination. For hypogeal germination, the hypocotyl will elongate. That's for the epigeal germination. While for hypogeal germination, the epicotyl will elongate. Okay. Apart from that, the cotyledon will emerge out of the soil in epigeal germination, while in hypogeal germination, cotyledon remains in the soil. Okay. Then for epigeal germination, cotyledons will turn green. That means it can photosynthesize. While in hypogeal germination, cotyledons do not photosynthesize. Okay, and then in the epigeal germination, the food or energy will be derived from the cotyledon. And in hypogeal germination, food or energy will be derived from the endosperm. So finally, let me talk about conditions that are important for germination. Number one is water. When we talk about water, water is needed for seed to swell. Water is needed for seed to do what? To swell. So, and it is when the seed swells that it can break. Okay, breaking of the seed is important so that the embryo will be able to emerge. Okay, and be able to grow. So this swelling will break the tester of the seed. So that is the importance of water. Then you have enzyme. What is the function of enzyme? The enzyme will help in transportation of simple materials to the embryo. Apart from that, it will also help in breaking or releasing or converting insoluble materials in the seed to soluble materials, insoluble food material to soluble food material, so that the seed or the embryo can make use of it while it's growing. Okay, that's for water, enzyme. Then the next one is oxygen. Oxygen is also needed for aerobic respiration to provide the embryo with energy. So this is a kind of aerobic respiration that will help the embryo to generate energy for itself. Then you also have warm temperature, and that is suitable for efficient working of the enzyme. As you know that enzyme work at optimum temperature. Temperature is too high, enzyme will become denatured. Temperature is too low, enzyme will become inactive. So warm temperature is needed for effective functioning of the enzyme. And lastly, you have the viable seed. That's talking about viability of the seed. The seed must be viable for the seed to germinate or for this plant to grow. And there are ways you can test for viability of the seed. Okay, so thank you very much. This is the last slide on this topic. I hope you've been able to learn important lessons here, and I hope it's going to help you as you prepare for your external and internal examinations. So please endeavor to like this video, share it with your colleagues, and then subscribe so that you can get educational content each time it is uploaded. Thank you for joining me once again. I really appreciate, and I see you in the next video.